Hi guys, welcome to Science Tech for Fun. This is Mr. Fasten here, and today I'm gonna show you something very interesting. Today I will be talking about the B-level 10 activity, investigating the effect of environmental variables on animal behavior using a choice chamber. Okay, so this is a co-protocol for biology, and this is the materials that we're gonna use today. Okay, so take a look. Right, so let's start assembling the choice chamber. There are infinite types of choice chamber. Okay, so the model I have here is made by Caroline, cell by Philip Harris. This is made of four chambers actually, and a central chamber, so 5 in total. The central chamber will connect the 4 ones. So basically what you need to do is take the center one and just slide inside. When you hear the click, it's fixed. Then you do it again with the other one. Click again, and the last. So this is my choice chamber made, and this is the lid for the internal chamber. Now I need to delimitate my environment areas. So we will work with 2 variables, light and humidity. Light, we need a source of light, so it can be a lamp. Okay, if you're lucky, it can be the sunlight. Lamp will do fine. We need aluminum foil to cover 2 chambers. For the damp or for humidity, I will need water, cotton wool, silica gel, and weighing balls. Okay, so how am I gonna delimitate? I'm gonna do 1 light, 1 no light. 1 light, 1 no light. And from the light, I will take 1 with water, the other one with silica. Silica will take all the moisture off, so the air there will be dry. The other ones will be 1 with water, 1 with silica again. Okay, so we're gonna have dark with water, dark with silica, light with water, light with silica. After that, I will keep my chamber for about 2-3 minutes to allow time enough for the vapor of water condensate and accumulate inside the chamber, and in the other hand, for the silica to start to absorb all the damp in the air and make the place dry. After that time, I will place my animal. Here we are using maggots, but the original design of this practical is with wood slice, but maggots are easy to find and way cheaper than wood slice, so work as the same. Okay, any kind of invertebrate, especially the ones that lives under rocks or in dark place, will work as fine. Okay, so let's start assembling. So the first thing to do is open my lids. I will open my 4 lids here, and I will choose 2 for dry, 2 for damp. Then I will place 1 plastic spoon of silica in each weighing bowl, and in the cotton wool I will soak it with water. This isn't the regular water, don't need to be distilled water. This is biology, so this is not important. The PPE that I'm using here, as you can see, is just gloves, okay? Is very necessary? No, for this practical, because these maggots I use it for fishing, so I didn't collect it from a cadaver of an animal somewhere, you know? So these maggots are not dirty or is not infected with any kind of disease. But some people don't find pleasant to deal with these animals, so gloves is a good solution, okay? I am fine with, but as example, I'm wearing gloves, okay? So now I will close my chambers. So close, you will see this lid have a little handle outside, so this show you which part is inside, which part is outside. So I close here, and then I will choose two to cover. So will be one dry, and one moisture. Okay? You don't need to worry if there is a little bit of light going inside the chamber. The big point here is to have as much as dark as possible, but don't need to be completely dark. Right, so what I'm gonna do now is I'm gonna open my central chamber and I will deposit my animals inside. The number of animals can vary, so you can either pick the animals, put in your hand and place inside, or you can just place as a spoon. The important part is the number of animals you place inside being counted, okay? So if you pick, you can pick 20 right here, or you can place a spoon here and count how many goes inside, okay? The number of animals is very important, so you can work with the percentage afterwards, okay? So, as I said, I don't have any problems, so I will pick this here, and I will count 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 16, 17, 18, 19, 20, 20, okay? So I will place my 20 here in the middle. Just check if they are not turning into poops. Okay? If they're turning to poop, they're not gonna be moving. So after it's placed here, I will put my light on the top and I will keep it for about 10 minutes, okay? So come close to see. Okay, so after 10 minutes, we can see that in this area here, I have 1, 2, 3. I have about 4, 5 here, 8 here, and let's see how many I have in the black with water and how many I have here with dry. So we can see that most of the maggots, they choose to go to dry and dark, then dry and light, third place moisture and dark, and the last one dry with light, okay? So this shows the behavior. So you can make the calculation how many it was in each chamber, and calculate the percentage of population, okay? Thank you for watching this video. If you like it, please give us a thumbs up. Please consider to subscribe, give us a comment, and see you next time.